So a pizza is divided into 8 slices, but there's 5 of us. Who doesn't get 2 slices? All right, this Norse Teaches Science today again with Mr. Norse, Biology Edition. And today we're going to talk about the cell cycle. We've been talking about it in the past couple videos. We talked about the background, we talked about interphase, but today we're actually going to get into the cell dividing. So by the end of this video, I can describe the steps that exist in the cell cycle, mitosis and cytokinesis. All right, so to begin, we already talked about interphases, prep phase, where it prepared to divide, but mitosis is actually the division of the nucleus, just the nucleus. And it's divided into 4 phases: prophase, metaphase, anaphase, and telophase. One way that you can remember it is this acronym, PMAT. It's the order it goes in. Actually, you can remember all cell cycle if you remember IPMAT-C. That's how we remember them in my class. But it's all up to you. Come up with a way for you to remember the order they go. All right, so let's talk about the first phase. So the first thing that happens during prophase is the chromatin turns into chromosomes. The nuclear membrane disappears. The nucleolus disappears. The centrioles move to the opposite ends or the poles, and mitotic spindle fibers form. So how it looks. So then I've got this dotted circle here. This represents the nuclear membrane disappearing. We're going to form, we're just going to have this cell have 4 chromosomes. Do you know how many chromosomes are in a human body cell? There are 46 chromosomes in a human body cell. 46. So you would have 46 of them right here, but we're not going to draw all 46. We're just going to draw 4. So we have the chromosomes formed. Remember, the chromosomes kind of look like X's. And then we have our centrioles at the poles. That's how I draw them. And then fibers coming off. And that would be kind of like what prophase looks like. After prophase comes metaphase. It's actually one of the shorter phases of mitosis. It's short because not much happens here. So what happens is the chromosomes are going to line up down the middle of the cell. In the centromeres, the middle of the chromosomes, are going to attach to the spindle fibers that run from one centriole to the other. So we're going to have our 4 chromosomes line up in the middle, kind of like what you see here. And one good thing is you should probably, if you're watching this, make sure you draw these out yourself because it will really help you understand. I know when I give a test in my class, I usually have them label each phase, so it would be good to have them so you can study. Have my centrioles and then the spindle fibers attaching. I know I don't draw that well. You try drawing on a computer. All right, so this is metaphase. After metaphase comes anaphase. What happens here is the chromatids are pulled to opposite ends of the cell. What's happening is they're actually splitting, the chromosomes splitting apart at the centromeres, since there's 2 chromatids, they're separating so that they go to opposite ends. Because remember, the result is 2 new cells, and each cell has to have the same amount of DNA. So it's got to rip the chromosomes apart. All right, so here's our chromatids. And if you notice, the cell is elongating. Do you know what that means? It means it's stretching out. So let's do the other side. And there is anaphase, cell elongating. All right, after anaphase comes telophase. if you can see, we're starting to see the two new cells. Not all the way yet. In telophase, nuclear membrane reforms. The nucleolus reforms. The chromosomes change to chromatin, and we're about to have cytokinesis form. So the nuclear membrane in both of the new cells reforms. And remember, it says chromosomes change to chromatin. So it's going to be stringy. Stringy. And this would be telophase. But as you can tell, they're not fully separated yet. That's what cytokinesis will do. Cytokinesis is where that's going to happen. So let's talk about that. In cytokinesis, you're going to divide the cytoplasm. But what's happening is the cell membrane is going to pinch close between the two cells, and you're going to have your daughter cells. So here's one cell, and here's the other cell, and they're going to have all their little organelles in here. I'm not going to draw all of them, because if I did that, we would be here for 20 hours, because I would be a perfectionist with it. And once the cells have separated inside of kinesis, they go back into interphase. It's a cycle. Each cell will interphase. It will go through G1, S, and G2, and then each of these cells would divide into two other cells. And that's how life keeps going on with the cell. So the result of mitosis is that there are two identical daughter cells. It means that they had the same number of chromosomes as their parent. So if you refer back, I started with 4 chromosomes in the parent cell, and each daughter cell had 4 chromosomes. But that is mitosis in a nutshell. There's a lot more to it. There's regulation enzymes and all this other stuff. So if you want to know more, go over to the Google it, learn about it, read a book. But I hope you learned something today. And this is Mr. Norris, and he is signing out.